Good evening, everyone. I hope I'm loud and clear. All right. So, how's your preparation going? Okay. So, Satyam says, great. What about Dilip? How's your preparation going? So, basically, Unit 5 is all about your developmental biology, right? And in your Unit 5, though 90% of the uh, things are from your animal dev bio portion, but there is a portion from your plant dev bio as well, right? So today I have taken up some plant dev bio sections for you now. Okay, so Satyam also says is going good, very well. So plant dev bio section, you get you get a lot of questions. Okay. uh, and you get both your part B questions and part C questions. Mostly there are part B questions, and like you will see, if I take the ratio, it will be 50 almost 50-50, or at least 50-40. Okay. Now whether it be part B or part C question, the question formats are absolutely the same. In part C only they will add some extra 2-3 lines and extend the paragraph. That's it. Okay. Now, so in today's topic, in today's session, we will discuss some few questions, and I will also explain the concepts behind those questions. Okay. So shall we start? Give me a thumbs up if we are all ready to start the question. Now, as we do it for all the other sessions, I will first show you the question, and then I will give you a 2 minutes time for you to go through, analyze it yourself, okay, and then we'll discuss. Because this is all about your practice, this is all about your revision. So I feel that you attempting first is more important. Okay. So, the first question. A researcher, while studying vernalization in plants, has made the following statements. Choose the incorrect one. Number one, vernalization causes stable change in the competency of the meristem to form an inflorescence in some plant species. Vernalization causes changes in gene expression that do not involve alteration in the DNA sequence. Flowering is not altered under normal growth conditions in plants defective in vernalization. And last, vernalization alters expression of FLC. Now, what is this FLC? This is your, what is this? FLC? So this concept of vernalization, it is also associated to your photoperiod. Okay. Now this photoperiod topic is placed under your unit 6, photoperiodism. And the gene regulation, that part is placed under your unit 5. So please, when you're going to study, club these two concepts together. Okay. Only then you will get the complete picture of this. Clear? Okay, Satyam says option 3. Dilip, what will you say? Try it. It doesn't matter if it is right or wrong, but trying is very, very important. Okay, because until unless you try, you will not know where, if, like, even if it is wrong, okay, if you don't try, you will not know where you're going wrong. And if it is correct, then very good. If it is correct, that means your concepts are all okay. So I always insist, try. Whenever you get a question, analyze whatever you're thinking. As per that, give me the answer. Okay, so basically, let me give you some information. What is vernalization? If I ask you, we're talking about vernalization, right? So, vernalization, you can call this a process where you expose the plants in cold, under cold temperature. You keep the plants under very cold temperature to induce flowering. Now, all the plants, they don't need this treatment. Okay, only certain plants needs this. So mainly what happens, it is seen that plants which are defective in vernalization, it can be because of some mutation or something, they often fail to flower. Okay, or maybe they show a very delayed flowering. The growth is normal, vegetative growth is normal, but flowering is getting delayed. Okay, and this is very vital for plants which need cold exposures naturally. Okay, you must have brought a plant from somewhere else which needs a cold temperature. Now if you want to grow it in a very hot humid climate, then you have to subject that plant to vernalization. Okay, now let's go into the statements. So the first statement is saying that vernalization causes stable change in the competency of the meristem to form an inflorescence in some plant species. Obviously, by the definition of vernalization, we understand that statement 1 is correct. Okay? Statement 2, vernalization causes changes in gene expression that do not involve alteration in DNA sequence. Now, this statement, this is also correct. Why? Because vernalization is mainly involved in epigenetic regulation. Okay, so the modification happens on your histone proteins, not on the DNA sequence. And then you have vernalization. All okay, flowering is not altered under normal conditions in plants defective in vernalization. No, this is definitely the incorrect statement. Vernalization alters the expression of FLC. So FLC full form is flowering locus C. And this is a major repressor of flowering in Arabidopsis. Okay, so if you look at this flower pathway, this is showing you a lot of pathway. I'll come to this. But for the timing, just focus on this part where vernalization is written. If you see here, you will see that basically this flowering locus C, FLC, this inhibits agamous. Okay, agamous like 20. And this protein is it has a very pivotal role. It simply transfers or transforms the vegetative state of plants to reproductive state by triggering leafy. Okay, now if flowering locus C is, so therefore flowering locus C is a repressor. Okay, now if this is present, so obviously it will repress agamous. Now low temperature that is vernalization, represses the repressor, that is flowering locus C. So then agamous is free, agamous is active. Okay, when agamous is active, it will help in the transcription of leafy. Leafy will come down and it will trigger the activity of all the other genes. So you will get sepals, petals, stamens, carpels, and all the four organs. ठीक है? So mainly this pathway is showing you the different ways by which agamous like 20 is triggered. Please excuse me. So there is light. Through light, there is photoperiodism. Okay. So different students, am I audible? Please, can you confirm? Students, am I audible? Okay, okay. So I hope you have understood the definition, like the regulation of photoperiodism. Or should I repeat? Okay, so like photoperiodism, you have the low temperature, that is vernalization, and like that you have the sucrose and gibberellins also. Okay. Now, coming to the second question. Uh, I hope now I'm audible. So what? See, the previous one was your part C questions, right? And this is your part B questions. So let us look at the part C questions. Okay, am I audible now? Okay, so what should be the answer of this one? The correct answer here, okay, before I come to the correct answer, let us look at this Arabidopsis. Okay, how the genes are regulated. So please can you confirm before I proceed whether I am audible or not? Can you give me a thumbs up? Okay, fine. So see, basically the four different walls of a flower. What are the four different walls? The outer one, the outermost is your sepal, right? The greeny parts that you get to see, the green things, that is your sepal. So if I draw a flower, okay, if I draw a flower like this is your sepal, the outermost. Now there are obviously not two, there are more than two, four, five, or six. So I'm just drawing two for the sake. It is all, it is in a, it's in a wall, okay? So circular. Now the next one will be what? The petal, the colored part. Okay, you have the petal, which is a colored one, right? Next, what you have is inside, you're going to have the stamens. All right, you're going to have the stamens. Now, stamen, if I draw, so stamen, if I draw it separately, this is your stamen. Stamen, this is your male part. Okay, so you have a filament and you have the anther. If you cross open this anther, you will get the 4 loculed anther with the pollen grains. Okay, you have in every locule, you have this uh, part. Okay, and the innermost wall is the pistil. Pistle. Okay, pistil. What is that? It looks like this. Okay, so if I draw the pistil, so this is your pistil, or sorry, carpel. Okay, you can call it as a carpel also. Sorry. Okay, carpel. Now, if you look at a carpel separately, you have the ovary, you have the style, and you have the stigma. And inside the ovary, you have the ovule. So your pollen grain falls here on the stigma, and the inner content goes right down and enters the ovule to fertilize the egg. That is the entire process of development. Okay. Now, basically, these walls, so I'm erasing it now. All right, I'm clearing the slide. So these walls, they are controlled by different set of genes. The set of genes has been placed in 3 different classes: class 1, class A, class B, and class C. Class A gene consists of apitella 1 and apitella 2. Class B gene contains apitella 3 and pistillata. Okay? And class C genes contains agamus. This gene. Okay? Only one. Now see. Class A gene is enough to form sepal. Look how I am marking. Okay, that is important. Class A gene is enough to form the sepal. Class A and B gene is required to form the petal. Can I make out 2 classes of gene are required here? Class B and C gene is required to form the stamen, and class C gene is required to form the carpel. So therefore, if there is a loss of C function, what will happen? This barrier, this boundary wall will be removed. So now the entire region will be occupied or by the expression of class A genes, will be filled with the expression of class A genes. So now what will you get? B is placed where it is. Okay. Only a sepal. AB petal. AB petal. Only a sepal. Similarly, loss of class A gene will have the entire portion filled with your C or dominated by C. Only C carpel, B and C stamen, B and C stamen, only C carpel. So the morphology becomes carpel, stamen, stamen, carpel. Okay. Next, if there is a loss of B gene, B class of gene, you will get only a sepal, only a sepal, only C carpel, only C carpel. Clear with this? Right? So now if I go back to this question. So now can everyone answer? Saritha has already answered. Satyam, Dilip, Josna. D. Joshna, Srishti, try. So they are saying that there's a loss of function of mutation occurring in B type. So that means you just have A and C, right? So what will you get? Yeah, this will be, there's a typing error here. This will be carpal. Okay? So the correct answer is sepal, sepals, carpal, carpal. Is that okay? Okay, I'm changing. Fine. Okay. Now coming to your apomixis. So they are saying following are certain statements regarding apomixis in plants. Apomixis cannot be used to maintain hybrid vigor over many generations in plants. In sporophytic apomixis, maternal genotype is maintained. There is an event meiosis during gametophytic apomixis and is also referred to as apomeiosis. In diplospory, meiosis of the megaspore mother cell is aborted, resulting in two unreduced spores, out of which one forms the female gametophyte. Which one of the following combinations is correct? So it's a total theoretical question. So apomixis is basically an asexual development of seed or embryo without fertilization, right? So we are saying asexual. So automatically it means there is no fertilization happening. Come on, this is very easy. Okay. So basically apomixis, uh, there is different types of apomixis, like you have the diplospory, apospory, and you also have the adventitious embryo, right? So diplospory is the situation where your embryo sac that is derived from the megaspore mother cell. Now you can derive that either by mitotic division or by interrupting the meiotic division. Okay. Now if you are uh, trying to get it from the mitotic division, so it is like mitotic diplospory basically. Okay. In that case, the megaspore mother cell will divide mitotically thrice and it will form 8 nuclei. Okay, but the embryo sac will remain unreduced and have the same number of chromosome and also your genetic material. If it is your apospory, then you have the nucellar cells giving rise to this embryo sac. We call it as apomictic embryo sac. Okay, and this is the most common type of apomixis in your higher plants. So in this case, you will see that this the initial cells, okay, they differentiate and they also undergo mitosis to produce the embryo sac. In fact, sometimes multiple embryo sacs also may be found. Okay, so in this case, the correct answer is statement B and D. In sporophytic apomixis, maternal genotype is maintained. Okay, and in diplospory apomixis, meiosis of megaspore mother cell is aborted, resulting in two unrelated spores, out of which one forms a female gametophyte. All right. Now, again, another question from the floral development of Arabidopsis. Now, when you are studying about the floral development of Arabidopsis, do pay attention on your anterenum also. The function of the genes are same, it's just that the names are different. Okay. So, which of the floral walls is affected by Apitella 3 pistillata mutants? Apitella 3 pistillata mutants. So, see, okay, first you answer, then I will give some comment. So, what should be the answer? No answer? Apitella 3 and pistillata, that means which one did I show you right now? It is your class B gene. If class B gene is absent, that means you just have A and C, right? So, class B gene is going to control which mutants? You had B here, right? AB gave you petals. B and C gave you stamen. So if B is absent, then obviously petals and stamens will not be formed. Right? Okay. Now you have two columns. See, this is again another part C question, but on the same topic. So can you see, like the topics as I told you, this very few topic. in plant dev bio section in that entire unit. Okay, but it's just that the questions are modified in different ways. Like if you look at this previous question, this is exactly the same question that we had done before, only it has been asked in a different way, isn't it? There also it was B mutant. So that is the thing. My suggestions: do not leave out dev bio, neither the animal part nor your plant part. So here they're giving you, given below are the list of proteins and their functions during floral induction. Okay, so they have given you flowering locus C, flowering locus D, flowering locus T, and frigida. Then you have column Y, which is giving you an activator of FLC, a mobile signal that induces flowering, regulator target genes that mediate the reprogramming of meristem to produce flower, a strong repressor of flowering. This is D, okay? A, B, and C, and D. So, can you make out which is the correct answer? Who can answer? I've already shown you that picture, so I'm not repeating it. The pathway I had shown you, and it is mentioned in the pathway. Okay, Sarita is saying option 4. Others? Like, even if you can match one, the rest will match. Isn't it? So the correct answer is option 4. Okay, see, flowering locus C, this is a strong repressor, isn't it? So A with 4, is there any option where you are getting A with 4? You only have it in option 4. Nowhere else. So even if you don't know about FLD, this FT and FRI, then also you will be able to get the correct answer. Okay, when you have the same uh, options more than once repeated, then it can be a bit confusing. But in this case, here only once they have shown that A with 4. Alright, so this is a repressor. Flowering locus D, this is regulator target genes that mediate reprogramming of meristem to produce flower. Flowering locus T, FT, this is a mobile signal that induces flowering. And you have the frigida, which is an activator of FLC. Okay, so you have to study the entire plant flower development. This question is coming from that section, not only just vernalization, but from the entire floral development, that part. Okay, now next question. Which of the following plant meiotic genes does not encode match domain transcription factor involved in floral organ specification? Very interesting. So basically we know that this floral developmental genes, these are all belongs to the madbox genes. But no, all of them are not uh, part of the madbox. So madbox is what? It is a conserved region basically. So who will give me the answer of this? Who's going to give me the answer of this? Match domain. So these are all transcription factors. The correct answer is Apitella 1. Okay. Apitella 1 does not have the match domain. All the rest, Apitella 2, Apitella 3, PI, AG, these are all your having match domain. So Apitella 1, leafy, agamus, they all function together. And Apitella 1 also functions here in the development of the floor walls. Okay, so it has a role to play in the transition from vegetative to your this inflorescence meristem to the floral meristem, and it also has a role to play in the ultimate transcription of the genes which are going to define the different walls. Okay? Now next question. So change in leaf morphology is observed during transition from vegetative to reproductive phase in several plants. The following statements are proposed to explain the above observation. Alteration in gene content of leaves of reproductive phase from those of vegetative phase. Differential methylation pattern of genes influencing leaf development and morphology. Mutation in transcription factor that prevents association with promoter elements of genes regulating leaf development, and small RNA mediated inhibition of gene expression of a homeotic gene. Which one of the following options represents a correct combination of statements that could explain the observed change? So, which one do you think is correct here? Anyone? Anyone would like to try this? No? The correct answer is B and D. That means this. So this is A, this is B, this is C, and this is D. So at the very first, I told you that in a different context, though, I told you that these changes that is happening, these are all epigenetic changes, right? So different differential methylation pattern of genes influencing leaf development and morphology. That is true. Mutation transcription factor that prevents its association with promoter element. No. Small RNA mediated inhibition of gene expression of a homeotic gene. Okay. So B and D are the correct statements. Because if it had been a mutation, it would have been a permanent feature. But here we see that it is only appearing during the transition, or it is like denoting transition. So it cannot be a permanent feature, right? So mutation is not correct. Alteration in gene content of leaves of reproductive phase from those of, no, this is not right. We are asking like what can be the reasons for this change? What can be the reasons for this uh, phenotypic change that we observe? So only B and D justifies the statement. Okay. Okay. Now, uh, this is, I guess you all know by now that we have this Lakshya batch going. So if you would like to join us and get some more explanations, clear all your doubts, your concepts, then contact us in this toll-free number. Okay, our team will assist you, provide you all the information that you need. Now, moving on to the last question for today. After this, again, you will get the link to the 5 questions. But before you appear for the 5 questions, you take up those 5 questions, revise, and then, okay? And if you would like to practice more such tough questions, then obviously you can check out the uh, Biotecnika app. Download the Biotecnika app, and then you will get the test T3 series. Okay, the tough test series. So you can give it, and you can obviously see your own performance and evaluate yourself. It is there for everyone, but it is there in the Biotecnika app. Okay, practice question sets. So here what do you see? They have given you a picture. Okay. So genetic screens from mutants affecting development of leaf trichomes have led to the discovery of genes regulating trichome patterning, especially trichome density and spacing. The following statements are made in this regard. Glabra 1 mutant plant will show fewer or no trichomes. Cells that form trichomes strongly express the glabra 2 and trypticon genes. Trypticon protein act as a positive regulator of trichome cell differentiation and the surrounding cells. Addition of exogenous jasmonic acid will reduce the number of leaf trichomes. You have to select the combinations with correct statement. Now, this is a question which you can't make any guess on logic. Okay, this is something that you have to know. So basically, the primary genes which control this trichome trichome development are your glabra 1, glabra 2, and your trypticon. Okay. Now this trypticon basically function as your negative regulator. Okay. So your glabra 1, glabra 2, they all promote this trichome production. So glabra 1 is considered as a central regulator of the development of trichomes, and it can directly activate the other genes, okay, which are needed for the development of trichome. Glabra 2, this is a transcription factor, and it also plays a very important role in trichome cell fate determination. Trypticon, this is a negative regulator and prevents the formation of all the neighboring cells. Okay, so if it prevents the neighboring cells, then it is actually helping in the spacing of the trichromes on the leaf. Okay, so it has been seen that mutations in any of the genes will obviously have an impact on the trichrome formation, but GL1 and GL1 plays a very important role. So what should be the answer? Saritha is saying option 1. Others? So the correct answer here is option 1. Glabra 1 mutant will show fewer or no trichromes and says that form trichromes strongly repress glabra 2 and trypticon gene. Why? Because they are the repressor. So they have to be repressed. Otherwise, the trichromes can't be formed. So A and B are the correct statements. Here they are saying it is a positive regulator. No, this is a negative regulator. And definitely this will not reduce. Okay. So that's all that I had for today's discussion. Do you have any questions? You can ask me. You will get the link. The link will get activated within 5-10 minutes. In the meantime, go through your notes. Okay? If you have any doubt, you can ask me. So, how do you think? Is this manageable? Unit 5 plant section, is it manageable? It is okay. If you have not studied this part ever, study it once. You will see how easy it is. Okay. So with that, I take a leave. Thank you, everyone, and meet you in another session. In the meantime, do practice, do revise. Bye bye, and good night.